this video, we discussed designing good experiments. This figure shows respiration rates of 10 frogs that were measured shortly after the frogs had been captured in the wild and brought into the laboratory. This is the pre-treatment measurements here. An insecticide was then added to the aquarium water of all 10 frogs at a dose similar to what the frogs would experience in their natural habitat. After 4 weeks of the frogs being exposed to this insecticide treatment, the respiration rate of the 10 frogs was measured again. So that's the after 4 weeks treatment over here. Please note that these were the same individual frogs measured in both categories. And this would be within subjects or paired design. Does it look like insecticide affects respiration rate? The height of the bars looks pretty similar. You would have to do a statistical test to provide adequate evidence, but even without a statistical test, we can guess that there doesn't look like any effect of insecticide on respiration rate in these 10 frogs. But is this a good experimental design? Has the experimenter controlled for all possible extraneous factors so that only the effective insecticide is being treated? Well, not exactly. If we look at the upper graph alone, the results suggest that the insecticide had no effect on the respiration rate of frogs. But the actual experiment included another set of 10 frogs that were not exposed to the insecticide, while everything else was the same. This is the no insecticide treatment shown as the white bars. Now we can see something more interesting going on. Frogs that were kept in the lab for 4 weeks without insecticide showed an increase in the respiration rates. Why might this be? It is possible that the first measurement had low respiration rates because the frogs were stressed from being captured and measured in the lab. But after 4 weeks to acclimate to lab conditions, maybe they were less stressed and their respiration rate increased. However, the frogs that were exposed to insecticide did not show an increased respiration rate after the 4 weeks, which might suggest they were still stressed because of the insecticide exposure. While it is important to look at the impact of the treatment in general, it is also important to have a proper control. In this case, in many cases, you need more than one control. In this case, there needed to be a control for the 4 weeks of time that passed. Time, or the order of treatments, can have an impact and must be controlled for. In this figure, we have the rate of oxygen consumption measured at 2 times, 0 and 90 minutes. At time 0, all animals were measured at 30 degrees Celsius. Then the animals were randomly split into 2 groups. One of the groups, the experimental one, was then subjected to a new temperature of 20 degrees Celsius, while the other group, the control group, remained at the original temperature of 30 degrees. After 90 minutes, the rate of oxygen consumption of all animals was measured again. Note the points here are just showing the mean of all animals within a treatment in time for clarity's sake. Letters A through D show some potential comparisons that can be made. Comparison D looks at the 2 groups at time 0 in the same condition to show that they all started the same. This helps show that there wasn't some kind of bias in assigning your animals into treatment groups, which if there was some kind of bias and they were different at time 0, that would add a confounding variable and would be a problem. So it's good to show that all the individuals in your 2 groups overall are similar at the starting point. Comparison C shows the impact within subjects just of time. So here we're just looking at the control variable. All animals were kept at the same temperature for both measurements at 0 and 90 degrees. So now we're just looking at the effect of time. Comparison B looks at just the impact of temperature, but does not control for the impact of time. So time can show things like what can happen over time is animals can acclimate or habituate or show some kind of learning or prior handling. There's a lot of things that can happen over time. Like even with a lot of our human experiments, sometimes the first time people are kind of nervous when you're measuring them, and then they're less nervous the second time or something like that. So you can have differences that just happen over time that isn't due to the treatment of interest, but due just to things that were different between the 2 time points. So if you compare C, that helps look at the impact of just those things that happen over time without looking at temperature. Whereas B is only looking at temperature and not taking into consideration any of those impacts of time. Comparison A looks at the impact of temperature without comparing it to the control, which would not control for the impact of time. It's just looking at temperature again, but it has a confounding variable of all the time stuff in there. So it's a little confusing if you just looked at A, right, and you didn't have a control, you wouldn't be able to know what the impact of just of time would be. So it is important to take into account the treatment impact of temperature or whatever treatment you're interested in, as well as time. We need to take into account the within and the between subject factors. So you have to look at what happens within subjects over time, as well as between subjects when you take the treatment into consideration. Let's consider what happens if we don't take into account the within subjects, which is also called repeated measures factors. If we have two treatments, A and B, and we just compare the mean of A, so you take all the individuals that had treatment A, you take the mean of that. So you're just taking the mean. So here's all our individuals, and then here's the mean and standard deviation of all individuals in treatment A. And then we do the same thing with B. So we take the mean and standard deviation of all the different individuals in treatment B. And then if we compare the mean of A to the mean of B, we might see that there's not really a significant difference because there's not a big change. But what you can see is there's a lot of individual variation. So how do we control for some of that individual variation? That there's a lot of differences between individuals. Well, we can actually, instead of taking the mean of everything in A and the mean of everything in B, we can use the paired design of this design and look within subjects at what we would call the repeated measure. So this individual, let's call it individual 1, was measured in treatment A and B. So if we take the difference between A and B just for subject 1, then we take that value as a difference. And then we take for subject 2 the difference between A and B. So now you've got a bunch of little differences. And then you take the mean of the differences. That's what's shown over here. So the difference between these is if you're doing independent samples where you're not taking into account the individuals are measured more than once, they're just independently taking an average of everyone measured at A and taking an average of everyone measured in B, and not taking into account that some of the individuals or all the individuals were measured in both treatments. So you're not taking into account the individuals. But over here, you're taking the difference between A and B for each individual. So you take the difference, and then you take the average of those differences. Now you can actually see that there is a measurable difference, that when you take into account all the within subjects aspect of this design, you do see a measurable difference in the treatment effects. So this is kind of like using each individual as their own little controlling for that individual variation. So doing a within subjects design or repeated measures or paired design, those are all kind of mean the same thing, can be a really powerful tool in trying to understand the impact of different treatments in organisms. Some things to keep in mind when you're designing an experiment. First of all, what are your treatments? How many treatments will you have? For experiments in this class, with our limited time and resources, and because most of you are newer to designing experiments, it is best to keep it as simple as possible. Try to limit to 2 main treatments, so treatment A, treatment B. How will you assign the subjects to treatments? It is generally best to assign subjects randomly to the different treatments, but there may be times when you have to do some non-random sorting to ensure some balance, because again, you want, when you compare among your individuals at your starting point or whatever, your pre-test, your baseline, that you want there to be overall no difference between your treatment groups. Because things can happen over time that aren't included in the treatment of interest, the order of treatments can have a huge effect. Why does the order usually matter? Because variables such as time of day, learning, fatigue, handling, stress, etc. can all have effects. How will you deal with this? So you need to think about possible effects of treatment order. What are some extraneous factors that might have an effect that you should try to control for? Some examples could include time of day or circadian rhythm. It's known that certain things like blood pressure or heart rate and things like that can actually change at different times of day. So you probably want to measure people at the same time of day, unless time of day is the thing that you're interested in and you want to see what the difference is in the morning versus the afternoon or something like that. Other extraneous things that are important would be stress, levels of stress, fatigue, time of last meal, time and amount of last caffeine, amount of exercise, amount of sleep, adjustment to measurement protocol, temperature, noise in the room, rest between trials. Do you need a placebo so that they don't know that they're getting the treatment? So there's a lot of things you need to consider. Size is also important, and having a larger sample size is generally better. You couldn't do the test on one individual and say you know anything from that result that you could say more widely about humans. Humans are so variable, so larger sample sizes help us reduce the influence of that variation. You need more individuals to get reliable estimates of what a typical individual in the population really is because we are so variable. If the data really are variable and you have a small sample size, the study may end up being inconclusive. However, getting enough subjects to participate and having the time to measure a large number of participants is not trivial. 3 is usually an absolute minimum sample size. Can't usually do statistics unless you have at least 3. But this, of course, depends on the test. But that sample size is really way too small for the kinds of tests we're doing. Conversely, a sample size of 50 is just not feasible because of the limited time we have, and it would be difficult to find that many volunteer participants. Think of what kind of sample size you could measure in about 3 weeks, noting that Mountain Day may take away one of those weeks. Think of other ways you can reduce variability and control for as many extraneous things as you can. Will you make all measurements within subjects, so the pre and post test on the same individual? Or will you make measurements between subjects, so the subjects are totally different in the different treatments? Because you can have each individual serve as their own control, within subjects or paired designs can sometimes get away with smaller sample sizes. You can still get interesting measurable impacts with paired designs. If you want to have totally independent subjects, then you tend to need to have larger sample sizes to help deal with the huge amount of variability among humans. So these are all things that you need to consider when you're designing your experiment.